And our next speaker is Edward Stroustrup from the Center for Genomic Regulation in Barcelona. Let's give him a warm welcome.
Is there a clicker pointer? Is it plugged in? Okay, cool. Can I plug it? Yeah, it doesn't seem to work. Okay, I'll use the keyboard. Okay, great. Just wanted to thank the organizers for getting me here, and also like Uri Alan and Max for getting the physics of aging biology session working, which was very exciting for me personally. So just a little bit of background to get us all on the same page. So aging, as everybody knows, involves a set of stereotypic phenotypic declines that move us from being young to being old and eventually dead. And unfortunately, these changes don't happen at a predictable rate because individuals vary. Everybody's aging at their own speed. And this ultimately produces a wide distribution of lifespan. Right? So aging not only makes life worse for everybody in general, aging also generates diversity. So if you look at a age-synchronous cohort of young people, people are relatively homogeneous. And over time, you start seeing increase in inter-individual heterogeneity. So aging is acting to generate inter-individual heterogeneity. And if you ask people on the street, your friends, why they think some people are aging better than others, those sort of three themes that pop up perhaps are the differences in our genome are making some people live longer than others. Perhaps differences in lifestyle, diet, exercise are going to have an impact. And many people will admit that perhaps there's some luck involved in the process of aging. So fortunately, we don't have to rely on our friends for advice. We actually have a pretty good body of evidence that would explain, at least in very general terms, why some people live longer than others. So twin studies and studies of heritability show that the heritability of lifespan is probably much less than 20%. So probably differences in our genomes are not explaining differences in our lifespan. There are many ways to go about asking the influence of lifestyle. I think the best, most direct way is to take animals out of the wild and put them into the laboratory, where we can control for lifestyle factors like diet and exercise and environment. And what you see here, I compare survival curves from humans on the left and C. elegans on the right, is even in isogenic populations held in very controlled conditions, you see a very wide distribution of lifespan, with some individuals living very short and some individuals very long. So I think in general, as the biggest factor generating inter-individual variability in lifespan, you can exclude lifestyle choices. That leaves us with luck. And to put it a little bit more professionally, the idea that variation might be intrinsic as an output of the biological mechanisms of aging. And so that's what my lab likes to study. And one thing that I'd like to tell you about today is some recent project and a method that we would like to use population asynchrony, these differences that arise over time between individuals, to map organismal networks and identify the mechanisms generating these non-genetic differences in lifespan. And this has been recently published, so I recommend to go to the paper. There's much more there than I can speak about today. So at the core, this is an experimental project involving development of a way to study gene expression data. So we spent several years, a couple years ago, really developing a high-throughput population-scale RNA-seq pipeline, inspired by some work that Simon Meloff and Ben Lerner had been doing a decade prior. And the idea is we take single individuals, these nematode worms, and we put them in lysis buffer, and we make individual worm soup out of each worm. And then we sequence it and get mRNA, full high-quality transcriptomes. And at this point, after a lot of optimization, we have the price down to about 20 euros per sample, which lets you go pretty big, especially if you're just a single research lab. And the output of this is a distribution of gene expression of each gene in the transcriptome across a population. Right? And so this is mostly work done by a three-way collaboration of our postdoc Matthias Eder, who's wonderful, and two very talented graduate students. So what we're doing is, what we'd like to see is this full trajectory of gene expression over time for individuals. So we can't get that. So this is a cartoon. So each individual has some gene expression, starts out high, and for this cartoon, it goes down over time. You have the individual trajectories, which we cannot measure because transcriptomics is destructive. But the idea is perhaps what we can do is we can sample cross-sections and then pull out the distribution of states that animals are occupying at particular ages, and then use those distributions to start trying to understand, infer the dynamics. And so, so what we get, sorry, there's a Mac, so the video gets messed up. But the idea is what we get for each gene is the mean expression across the population and then the variance. And these two quantities actually were quite hard to get right because when you're looking at differences between individuals, you're also looking at differences between samples. And so we really had to get down into the guts of the sequencing technologies to understand what aspects of the noise we were measuring are technical versus biological. And so there's a lot of supplementary data there if you're ever curious. But the result is an atlas that we trust pretty well, and it's a population scale atlas of gene expression variation. So on the x-axis, you have mean expression. Each point is a gene. And so if you've ever looked at transcriptomic data before, you'll be familiar with the concept that there's a very wide dynamic range of expression levels. So the most abundant gene in C. elegans cells and in your cells are all a million times higher abundant than the lowest expressed genes. And what we were interested to see was that there's a similar, there's a wide dynamic range in gene variances. So some genes are held very tightly controlled across a population. So all the individuals are very similar. And so for other genes, there's a much wider distribution. So you can look at how aging affects this accumulation of gene expression variation. Again, this is a plot. Each point is a gene. At x-axis, we have the change between day 1 and day 8 of adulthood, so young and old, in mean abundance. And on the y-axis, you have the change in variance. And we've colored genes based on how they're changing, right? So you have this category of genes that are going up with age and are becoming more variable. You have genes that are going down with age and becoming more variable. And we actually see very few genes on the lower side of the axis. So basically, the whole transcriptome is becoming more variable. But what I'm actually most interested in, was most surprised to see, was this category of genes that aren't changing, aren't really going up or down in mean expression, but they're going up a lot in variance. Right? And so if you quantify the number of genes in these categories, you actually see that almost half the transcriptome is increasing in inter-individual variation without changing the mean. And so I think this is kind of like a hidden aspect of aging, a much larger subset of the transcriptome that's showing age-associated changes that people haven't appreciated. And people have missed it because if you're doing bulk transcriptomics, pooling populations of people, of course you're averaging over this, you'll never see it. In clinical research, using people, you actually have lots of individualized data, but I think most people would assume that this variance has something to do with environment or genetics, and I think we're showing that this is actually an intrinsic generation of the aging process. And finally, single-cell methods are going to conflate intercell variation from the inter-individual noise, and so they're also going to misattribute it. So we were excited to see interesting things going on with gene expression variation. But one of the strengths of C. elegans is you can actually get down to the mechanisms and try to look at phenotypes. So we started trying to figure out where this gene expression variation was coming from. And as most of you know, C. elegans has differentiated tissue types that are somewhat analogous to humans. And there are subsets of genes discovered by Olga Troyanskaya and Colleen Murphy that are only expressed in certain tissues. And what we did is we started looking at those as a way to pick out tissue scale changes. And so what we have here is the correlation plot of genes in the x and the y axis, and then the correlation across the population in their expression levels, grouped by the tissue of expression. And so you can see that the germline genes are all highly coordinated. So you have the red is highly is positively covariating genes, and so is the somatic genes, the hypodermis and intestine. And what you see is an apparent anti-correlation in between the germline and the soma. And so if you collapse this box into just a distribution of the gene pair correlations, you see this bimodal distribution of genes that are expressed within the same tissue and then genes that are anti-correlated across germline and soma. And so to try to get to the bottom of this, we spent quite a bit of work trying to do absolute quantification with ERCC spike-ins. And what we found was you can quantify the size of a tissue, at least the size of the tissue-specific transcriptome, by summing up all of the tissue-specific transcripts. And what you see in youth in day 1 is that some individuals are larger, some individuals are smaller. But the size of the somatic transcriptome relative to the germline is apparently very correlated and perhaps highly coordinated. Over age, this coordination breaks down, and you start seeing animals with a much higher variation in their relative sizes of these two tissues. And so the idea is perhaps it's the variation that we're seeing at the gene expression level is actually a result of a tissue scale phenomenon. So we went to check that experimentally. So in C. elegans, you can actually ablate the germline. Don't know how to ablate the soma yet. But the idea is you can make this mutation, you get a population, you rebuild your single individual expression atlas using animals with other germline. And you can see this completely eliminates this positive and negative correlation structure you see, demonstrating directly that most of the gene expression variance you see at the organismal level is tissue scale phenomenon. Another way to look at this is to do the PCA plot of gene expression, and you can see in black the intact animals are very variable, and that collapses in the germline ablated. Right. So another nice thing about C. elegans is you can quickly go to phenotype, and so people have known for a long time, starting with Judith Kimball 2 decades ago, that when you ablate C. elegans germline, you extend lifespan by a lot. And when we loaded these onto the lifespan machine and did the experiments very carefully, we saw that germline ablation was extending lifespan while reducing lifespan variance, which is something we hadn't seen before. And when you run an AFT regression model to try to normalize for timescale, it becomes very clear that what germline ablation is doing is rectangularizing the survival curve. Right? And so the relative change in variance, independent of timescale, is almost 50% lower variance. Right, so that was very exciting. Put us in a model where you have a worm, and there are 2 distinct transcriptional units, the germline and the soma, and they're varying partially independently from each other. And this variation is somehow related to variation in lifespan, because when you remove the germline, you remove the lifespan variation. Right? So we were excited to have some sort of feature that was explaining midlife physiologic heterogeneity. But germline ablation is kind of a big hammer to take to an animal. So you're wondering if there's a more surgical, maybe we could get down to some genes that have more precise effects, right? So we wanted to look for genes whose activity is contributing to organismal gene expression variance, right? And then counter screen those genes for genes that might then be driving lifespan variance. Right, so we didn't actually have the resources to do RNAi screening of single worm transcriptomes. It's actually a lot for a single lab. But we thought for a while and realized that it should be possible to find the genes we're looking for using bulk transcriptomics. And I'm not going to go into the details of the transcriptome models. I'll summarize them. The idea is from the average effect of a gene, what it has across a population, perhaps individuals are varying along the same axis that you can capture with the average effect. And the cool thing is that the average effect we can measure pretty quickly with our sequencing method, and we already have our atlas. So using our atlas, we can guide rapid screens, and we can go sort of like 200 times faster. And so I'm throwing this up on the board, really in case anybody watching this online who wants to pause the video and is interested in the statistics can take a look, and I'm happy to answer questions after. But the long and the short of it, this is like not an aging clock. It's a different way of thinking about how individuals vary, not projecting it over time, but projecting it over an intervention. So we had a method that we thought would work, and so we picked some candidates to screen from our atlas, looking at genes that are highly variable. Also looking at network connectivity using various graph methods to find genes that we thought might be central to gene expression. And we selected 90 targets across all tissues for a first screen. We took animals, put them on the RNAi, we aged them, and then we collected transcriptomes. And what we found is that there's a great diversity of gene expression effects. These genes aren't all having the same effect on the transcriptome. So this is a hierarchical clustering. All of our targets are put in this hierarchy, and you can see at least 7 independent groups of very different gene expression effects. But when we started asking what fraction of gene expression variance these targets could explain, we were very happy to see 40 of our targets were hits that individually could explain more than 10% of gene expression variance. And we're very excited to see 12 genes that individually would explain more than 30%, some as much as full genome germline ablation. Right, so then we started counter-screening our gene expression hits for lifespan phenotypes. And we were really surprised to find that all of the ones we tested, we couldn't test all of them, but the ones we looked at, they all had lifespan phenotypes. Right? And so I'm not familiar with that many screens that would produce that kind of enrichment, although I don't know for sure what the null hypothesis should be. But the result is that they all affect lifespan, right? Some of them we already knew, like knocking down ribosomes, ATP components, but it was nice to see. So then we counter screen for lifespan variance phenotypes. And we found genes that extend lifespan, like this small peptide signaling molecule, but also produce a rectangularization, reducing lifespan variance. And we found genes that would shorten lifespan and similarly rectangularize the survival curve. So this is a plot of the effect on variance. Remember, full germline ablation reduces variance by about 50%. And so some of our targets, knocking down single genes, are having as much of effect of germline variation. And I don't show the data here, but our top targets are actually having no effect on reproduction. So we're quite excited to see that. So we arrive at a place where we can start asking general questions. Why do some individuals live longer than others? And I think part of the answer is that it's the intrinsic activity of these particular genes that we're now finding. And the cool thing is that these genes are not just in the germline, they're distributed across all of the tissues we've looked at. And it's only a subset of the ones that we've actually phenotypically validated. There's many more on our list. And so we can refine our model. No longer it's a soma in the germline, but it's all of the different tissues that are interacting with each other. And there's targets in each of the tissues. And we actually can find them, and they have names. But for the purposes of where we are now in the project, the specific names don't matter. We're still trying to understand how they interact physiologically. But what's important is they're all contributing to gene expression variance and lifespan variance. And so it seems like the reasons why people live longer than, why some individuals live longer than others is because of a large number of mechanisms distributed across the animals. But the cool thing is we know how to find them, right? So many mechanisms are intrinsically generating age-associated heterogeneity. The germline is transducing much of that heterogeneity into lifespan variance. And we have an approach that can systematically identify these types of mechanisms described in our paper. And I think there's a broader translational point, which is actually one of the reasons I was really excited to come talk to you guys, which is that the fundamentals of this is not like a worm-specific type of project. We're just going on trying to measure how individuals are differing from each other and then project that on to the effective interventions. So we have a systematic approach that we think can identify new types of targets that are aimed at making lifespan fairer. And just to end on a philosophical note, I think that there's lots of reasons why everybody would like to live longer, but I think there's also lots of reasons why everybody would like to live, as long as their neighbors, right? To have everybody reach their maximum potential, right? And so now I think we're getting a look into some of the mechanisms that are taking what would otherwise be a homogeneous population in respect to chronological age, and then generating the differences that then are reported as differences in biological age over time. Thanks.
Thanks for an awesome talk. We have time for a question.
This really is very amazing talk. So what you're saying is this stochasticity transcription is controlled by genes in part, and which in turn is related to lifespan. So I was wondering, if you look at those gene ortholog in humans and look at genetic association with a disease or lifespan?
So control is a strong word. I think we might not be watching the control of noise, but rather the propagation of noise as it starts small and then becomes organismal. And no, we have not looked at the translational things. Something we should do.
And I just wonder if like DAF2 or interventions will affect those transcripts as well, upstream.
So that's figure 6 of the paper. So we did all of this stuff in DAF2 as well, and we're really surprised to see very few effects of DAF2 on inter-individual variation. And DAF2 is epistatic. So when you reduce lifespan variation with GLP-1, and then remove DAF2 from the system, they live long while scaling the now lower variance lifespan distribution. So we can talk more about what that means, but DAF2 seems to be not what we're studying. It's sort of orthogonal to it in an interesting way.
Hello. Nick? Yeah, over here. Here, yeah. Yeah, hi. Great talk. It's really beautiful. Do you think that this heterogeneity actually reflects some adaptive process that predates aging and maybe selected for in evolution?
Yeah, so there's a whole bet hedging theory of evolution where the idea is you generate physiologic heterogeneity so that some animals happen to be in the right physiologic state when starvation comes or things like this. So it's totally possible that there's some selective purpose for all of these mechanisms. I think I'd like to understand the mechanisms themselves a little bit better before we start talking about their purpose, because in the end there's variation in the timing of bridge collapse. So you can generate heterogeneity without any kind of selective forces, and so maybe we'll know more in a bit.
Okay, we don't have time anymore. There's lunch now, and please remember at 12 o'clock the startup pitch session starts here. Thank you.